Okay, we have a biology paper, February, March 2023, paper 4-2. So let's start from question number 1. Number 1A says, figure 1.1 is a simplified diagram of the circulatory system in humans. Alright, so here is the diagram of the simplified circulatory system in humans. As we can see, we have different parts on it, and we have label lines together with letters. Okay, we have organs starting from the head, heart, lungs, we have the liver, we have the stomach here, and have the intestines together with other organs. Now, the questions say, state the letter from figure 1.1 that represents the blood vessel that contains blood with the highest concentration of oxygen. Alright, so blood with highest concentration of oxygen will definitely be blood coming from the lungs, where gas exchange is taking place, and going back to the heart. So from the lungs back to the heart, it will definitely be this part B here. Alright, so blood vessel B is the one that will have lots or high concentration of oxygen. We'll label B. And then state the letter from figure 1.1 that represents the hepatic portal vein. Hepatic portal vein, this is a vein that connects the intestines to the liver. Alright, so when we come back to our diagram over here, it will be this vessel over here. Okay, it carries the substances from the intestines over here to the liver, and definitely to be represented by vessel C, so light C. Okay, then state all the letters that represent arteries. That arteries carry blood away from the heart. Alright, so we'll look at arrows which are pointing away from the heart. Alright, they'll be representing arteries. Alright, so we have this one here, which is labeled as A. We can see it's definitely going away from the heart. We have this one here, D. It's definitely going away from the heart. We have this one here, E. It's also going away from the heart. So you can see A is away from the heart, E is away from the heart, D is going away from the heart. So all these could be representing what? Arteries. So say A, D, E. These ones here represent arteries. Alright. Roman 4 says, explain the importance of the part labeled Y. Now we need to identify the part labeled Y. Yes, we can see it. The part labeled Y is here in between the chambers of the heart. Now this part is usually known as the septum. Alright. The septum separates the left side of the heart from the right side of the heart, whereas the left side of the heart is usually having blood filled with oxygen, while the right side of the heart has blood filled with what? With carbon dioxide. So in short, the septum prevents mixing of oxygenated and deoxygenated what? Blood. Alright, yeah, so we can say the part Y is septum. Alright, it separates the right and left side of the heart. Okay, and left side of the heart. That's the first thing. But also it prevents mixing. Of oxygenated and deoxygenated blood. Alright, yeah, so this is what it does. It prevents mixing of oxygenated and deoxygenated blood, but also it separates the right side from the left side of the heart. Then Roman 5 says, state the evidence from figure 1.1 that the diagram shows a double circulation system. Now when we speak of double circulation, it means that the blood is flowing through the heart twice. Okay, or we have two loops of circulation. And of course, when we look at our diagram over here, we actually have two loops of circulation. Alright, we have a loop, this one here, which is entering through blood vessel F. Okay, this is a loop for the oxygenated blood. And then we have this one here, which is leaving through blood vessel A. This is a loop for oxygenated blood. Alright, so we can say that it proves that it is double circulation because blood is flowing through the heart twice, and we have a loop that is filled with oxygen, and we have a loop that is filled with what? With carbon dioxide. So here we'll say that blood flows through the heart twice. Alright, so blood is flowing through the heart twice in two loops. A loop that is filled with oxygen, known as systemic circulation, and a loop that is filled with carbon dioxide, which is known as pulmonary circulation. Alright, we move forward. B says figure 1.2 shows the pressure of blood flowing through different blood vessels as it travels around the body. Venules are narrow vessels that connect veins to capillaries. Now we have our diagram over here as we can see it. Alright, we have a part labeled arteries, we have part X, we have capillaries, we have venules, and we have veins. Alright, then the question says blood vessel X in figure 1.2 supply blood to skin surface capillaries and have a role in maintaining constant internal temperature. State the name of blood vessels that are represented by the letter X in figure 1.2. Alright, now arteries, they usually branch to form something known as arterioles. Alright, so blood usually flows from arteries, then it goes to arterioles. Then from the arterioles, that's when it goes to capillaries, venules, then veins. So the part labeled X over here represents arterioles. Alright, yeah. And then Roman 2 says explain the reasons for the changes in pressure in the arteries. Alright, so what could be the reason of changes in pressure? You see in the arteries over here we have like a rise and a fall in what? In pressure. Alright, so the changes in pressure in arteries could have resulted from either contracting or relaxing of the ventricles and atriums within the heart. Okay, so when the ventricles contract, it means the blood is being forced out of the ventricle and out of the heart. Right, then there the pressure will do what? Will increase. And when the ventricles relax, the pressure is going to do what? Is going to fall, because when the ventricles relax, it means they are opening up and blood is rushing into the ventricles. So we can say here, say pressure changes due to contraction and relaxation of ventricles. So pressure changes due to contraction and relaxation of ventricles. Pressure changes due to contraction and relaxation of ventricles. When ventricles contract, pressure increases. Alright? So we'll say here, when ventricles contract, pressure increases. Pressure increases, and when they relax, pressure decreases. Alright, yeah. So this is what is happening in case of alteration of pressure in the arteries. Okay. Using the information in figure 1.2, explain the structural adaptation of arteries and veins. Okay, so how actually the structure of a vein enables it to function. Same goes to the arteries. Now, arteries, first of all, they pump blood with a very high pressure. Why? Because they have thick walls. Alright, so that is one adaptation. Arteries have thick walls to withstand high blood pressure, but apart from that, they also have narrow lumen to withstand what? Pressure. So we'll say here, arteries have muscular muscular, or you can say thick walls to withstand pressure. That's one thing about arteries. But also arteries have narrow lumen, or you can say small lumen, to maintain what? To maintain pressure. To maintain high pressure. But also they have elastic walls to allow what? Stretching and recoiling as pressure is increasing and decreasing. So again, they have elastic walls to allow recoiling and stretching. And then for veins, we also say that they have wide lumen. Why do they have wide lumen? Because blood is flowing with low pressure. So veins have wide lumen, yes, as blood is flowing at less resistance. As blood flows with less pressure or less resistance. All right, yeah, that's the first thing. But also they have valves, because the blood here is not flowing at a high pressure. They have valves to prevent backflow of blood. So veins have valves to prevent backflow of blood. Okay, yeah, so these are the structural adaptations that we find in arteries and veins, and they bring about the functions of these parts. All right, yeah. And then we move to the next part, that a student investigated the digestion of starch. Figure 2.1 shows the apparatus she used. So these are the apparatus she used in her investigation. All right, we have test tube A and test tube B. In test tube A, there was a dialysis tubing bag placed inside distilled water, and inside the dialysis tubing bag was starch solution. Same for test tube B. The test tube was filled with distilled water, then a dialysis tubing bag was placed in it, and of course this bag had starch solution and amylase in it. Then the question goes further to say dialysis tubing is used to represent a cell membrane. Right, so it's partially permeable, just like a cell membrane. The dialysis tubing material allows small molecules to move across it, but not larger molecules. Test tubes A and B were set up as shown in figure 2.1 and placed in a water bath at 7 degrees centigrade for 30 minutes. The liquid outside the dialysis tubing in test tubes A and B was tested with Benedict solution at 0 minutes and after 30 minutes. Table 2.1 shows the results. So here we have the results of whatever is taking place for both test tubes. At time 0, when Benedict was added into the samples from A and B, it remained blue. But after 30 minutes, again, when Benedict was used to test, for test tube A, it turned blue, but for test tube B, it turned red. Alright, now the question says, using the information in 2.1, figure 2.1, using the information in figure 2.1 and the table 2.1, explain the reasons for the differences in the results for test tubes A and B. Alright, so for test tube A and B, what should be the reasons? Let's start with test tube A. We can say that at time 0 it was blue, but after 30 minutes it was still blue. And when we come back to our sample here, we find that we only had starch solution in the dialysis tubing bag. Alright, starch is a large molecule. Okay, when tested with Benedict solution, it will retain the blue color. Okay, because this starch is a large molecule that has not been broken down. But when we come to test tube B, the starch solution was treated with amylase. Now amylase will break the starch solution into its simplest form, which is glucose. Glucose, when tested with or when reacted with Benedict, because it's a reducing sugar, it will turn into what? Into red. And this is the reason after 30 minutes, test tube B turned red, but for test tube A, it retained blue color. So we'll explain here. Okay, So we'll say test tube A retained blue color after test with Benedict after 30 minutes because starch is not broken down. Starch is not broken down. Okay, then in test tube B, the sample turned red after 30 minutes as the starch is broken down into simple sugars by amylase enzyme. Okay, by amylase enzyme. All right, yes. Then amylase from the tubing bed diffuses into distilled water. Diffuses into the distilled water or into the test tube. Into the distilled water or into the test tube. All right. By what? By diffusion. Okay. So after it is broken down, it becomes a small sample, and this small sample, after it is broken down, it becomes a small sample, and this small sample diffuses out of the test tube into the what? Into the out of the dialysis tubing bag into the test tube. All right. Yeah. And then for test tube A, there's no breakdown of starch, so even when we tested it with Benedict solution, it retained the blue color because there was no any kind of breakdown. And of course, starch is not a reducing sugar, so there's no way to turn blue in presence of what? Of Benedict's solution. All right. Yeah. That was for that question. Then we go to B. B says complete table 2.2 by writing in names. Complete table 2.2 by writing in the names of missing enzymes, substrate, and products. So we have to complete this table by stating what is missing, if it's an enzyme, if it's a substrate, or a product. Alright, so we have an enzyme known as pepsin. Pepsin is an enzyme that acts on proteins. So the substrate here is going to be a protein. Okay, now what is the product of breakdown of protein? Whenever proteins are broken down, we get amino acids as the products. Then the second part, we have been given products which are known as fatty acids and glycerol. Now these are the smallest units of what substance? They are the smallest units of lipids. Alright, so the substance is definitely a lipid. When broken down to give us fat acids and glycerol. Now what enzyme breaks down lipids? It's usually lipase. Alright, yeah. Then we have trypsin. Now trypsin is an enzyme that acts on proteins also. So the substrate is going to be a protein. But what will be the product? The product is going to be an amino acid. Then we have glucose as a product. What is a substrate when we have glucose? When we have glucose, substrate can be starch and enzyme can be what? Amylase. Or substrate can be maltose and substrate can be substrate can be maltose and enzyme can be what? Maltase. So we have different substances. We can say starch is the substrate and amylase is the enzyme. Alright, or you can use maltase as the enzyme and you can use maltose as the substrate. Okay, yeah. C says state the name of structures that increase the surface area for absorption in the small intestine. Now, structures that increase surface area for absorption in the small intestine are usually microvilli, which you find on the what? On the villi. Right? So we state the what? The microvilli that we actually find on the villi. Alright, move further. Question 3. Question 3 says, figure 3.1 shows part of a classification diagram for plants. Complete the diagram, the figure 3.1. Alright, we can see the figure that we have over here. It has plants, then here we have, ferns, then on the opposite here we have a blank. This blank branches to form monocotyledons and another blank over here. Now, monocotyledons are a branch of flowering plants. Alright? Monocotyledons are a branch of what? Of flowering plants. So it means up here, this first blank here is for flowering plants. And it makes sense because next to it is a fern. A fern falls into the non-flowering plants. Okay, so flowering plants can be monocotyledons or dicotyledons. So the second gap over here is to be filled by dicotyledons. Alright, yeah. And then we move to Roman 2, which says, describe 2 identifying features for monocotyledons. Now, what are the features that are used to describe something as a monocot? First of all, we have to know that they have, when you see their leaves are narrow. Okay? They have narrow leaves. So here we say that monocotyledons have narrow leaves. But also we have like vein-like structure inside the leaves. The veins for a monocot are parallel. Alright, so we say also they have parallel veins. Yes, so this is how monocotyledons look like. Or those are some features for the monocotyledons. Alright, B says, figure 3.2 shows the life cycle of a fern. The life cycle of a fern has some similarities and some differences compared with the life cycle of other plants. The letters represent processes that occur during the life cycle. The haploid stage of a life cycle is represented by N, and the diploid stage of a life cycle is represented by 2N. Alright, so we can see in our diagram here, for example, the sporophyte, it's 2N, so of course it involves a diploid stage. Then we have A, process A, which results into spores that are existing as N, which means they are in haploid stage. Then B, then we have the adult gametophyte, which is existing in haploid also. Then we have process C, which results into formation of gametes, which are also haploid. Then after that we have D, then we have zygote, which is in state of diploid. Alright, yes. And then Roman 1 says the adult sporophyte has 1,200 chromosomes in its body. Set the number of chromosomes in the body cells of the gametophyte. So the adult sporophyte is like here, okay, and it is represented by 2N. So it means that 1,200 is 2N. But when we say the gametophyte here, gametophyte has N, it means it has been reduced, or it means the number of chromosomes has been reduced. How so? It has been reduced to half, because we had N, now we had 2N, now we have only a single N. So it has been reduced to half. Now, to get the number of chromosomes in adult gametophyte, it means it's equivalent to saying 1,200 divided to 2. Alright, when we simplify this here, will give us 600. Alright, so the number of chromosomes in adult gametophyte will then be 600. Alright, then this identify and describe process A. What is process A? Alright, so process A is here. Alright, we have 2N from adult sporophyte, then we have spores that exist as N. Now this process A involves halving the number of what? Of spores, okay, or reducing the number of what? Of chromosomes. This process which reduces number of chromosomes is known as meiosis. So we'll say that process A is meiosis, okay. Yeah. Okay, what does it do? So we'll say here meiosis is a cell division that halves the number of chromosomes. That halves the number of chromosomes. Meiosis is a process of cell division that halves the number of what? Of chromosomes. Alright? Yeah, that is the description. We have identified the process that it is meiosis, and what it does is halving the number of chromosomes. Alright, now the importance of meiosis is that it forms gamete cells, alright, but apart from that, it forms gamete cells that are genetically different. They look different from their parent cells. So here we'll say that it produces cells that are genetically different. Okay, they don't look the same. That is meiosis, what it means, and of course the advantage. Alright, so Roman 3 says state the name of the process D. Okay, so we come back and look at process D over here. When we look at it, we'll see that we had two gametes which existed as haploids, and then after that process D occurred, and then we had a zygote which existed as a 2N. So this means that the two gametes fused together, and the name of this process will be fertilization. Alright, yeah, so that is the name of the process. Okay, C says figure 3.3 shows the paths of two flowers from two different plants of the same species. So two flowers from two different plants, but it is the same kind of species. Now what is meant by the term species? So we say that species, these are organisms that can breed to reproduce fertile offsprings. Alright, so they are organisms that can breed to reproduce what? Fertile offspring. They must be fertile. If they are not fertile, then the two organisms that were involved were not what? Were not of the same species. Alright, so we say that a species is an organism that can reproduce to form fertile offsprings. Alright, the offsprings must actually be fertile. Roman 2 says annotate figure 3.3 to show the mechanism of cross-pollination by labeling the structures involved, drawing an arrow to show the pathway of pollen. Alright, so the pathway of pollen will actually be from the anther to the stigma of what? Of the flower. So let's assume it's coming from this anther to this stigma over here. Alright, this part here will be the anther, and of course it's going to produce the pollen. Alright, and this part here will be what? Will be the stigma. Alright, which is going to trap the pollen. Okay, then after the pollen have been trapped on the stigma over here, they will grow through a pollen tube down the style until they reach the ovary and get to fuse with what? With the eggs inside the ovary. That is how cross-pollination occurs. Okay, draw an X on figure 3.3 to show where fertilization will occur. Now, fertilization will definitely occur inside the ovary of the flower. So here, inside the ovary is where fertilization will occur. Okay, explain the disadvantages of cross-pollination compared to self-pollination. Now, cross-pollination involves movement of what? Of gametes from one flower to another, but flowers of the same kind of species. Alright, so one disadvantage is that it depends on pollinators. Alright, so it depends on pollinators, meaning that without pollinators there will be no transfer of pollen. Alright, yeah, so it depends on pollinators, but also it is less successful. Okay, because yes, pollen may be carried, but we can never be certain that the pollinator will definitely move from one flower of this kind and then land on another flower of the very kind, of the same species. Alright, so we can say that it is less successful. Okay, meaning that it has less chances of being successful. But also we need more pollen. It needs a good number of pollen because some might be waste. Okay, yeah, so we say here requires more pollen. It requires more pollen, but also it needs more than one plant. Requires more than one plant, right? From one plant to another. But also it requires energy. So here requires what? Energy. Okay, so that is the thing with cross-pollination. It depends on pollinators, needs a lot of pollen, requires energy, and of course it is less successful. Alright, we go further. Now we are on to question 4. It says here, figure 4.1 is a photography of a tree that has been gilded. Gilding involves removing a complete circle of a bark of foam from around the tree. As we can see, a part of foam has been removed. Explain why the area above the gilder in 4.1 will become swollen. With this gilding process, we'll remove the phloem, alright? And the phloem is involved in transportation of the products of photosynthesis. So it's involved in transportation of sucrose and other products of photosynthesis. Alright, so when the phloem has been removed, it means that there's nothing that can carry the products of photosynthesis from the upper part of the plant, which means the sources, to the lower parts of what? Of the plant. Now what will happen at the end of the day? This upper part, which has the phloem, will now appear to swell. Alright, so use the term source and think in your answer. Okay, so we're going to say that sources, the process is a place where photosynthesis is occurring, and it's definitely the leaves. And of course the roots are going to be acting as what? As the sinks. So here we say that the products of photosynthesis from the sources or leaves are carried by phloem to the sinks, whereas here we know that sinks represent what? Roots. Okay, they are carried by translocation process. Okay, so they are carried by translocation process. Right, now what will cause the sinking? The removal of the phloem, the removal of the part of the phloem stops the translocation process, causing the part above the gilder to become swollen. Alright, so this is the case. So when we remove the part above the gilder, it will inhibit the process of translocation, and of course at the end of the day, the part above the gilder here will appear swollen, while the one below it will not appear swollen. Alright, because now the substances for photosynthesis are not carried through anymore. Okay, explain why leaves in gilded trees are still able to receive mineral ions from the roots. Alright, so the thing is, the phloem is usually at the outer part, while the xylem is in the inner part. So the leaves will still be able to receive minerals because it's only the phloem that has been what? Has been removed. Alright? Only the phloem has been removed while the xylem is still there. So we'll say here that transports minerals is further inside the trunk. Yeah, so this xylem that transports minerals is further inside the trunk, hence the transportation will still go on for the case of minerals. Alright, explain why glucose made during photosynthesis is required for the absorption of mineral ions by the roots. Alright, so glucose is needed because glucose is a component necessary for respiration. Alright, and when respiration takes place, what do we get? We get ATP energy, which is used in the active transport for the absorption of what? Of minerals. Alright, so we'll say here glucose is required for photosynthesis. Alright, glucose is required for photosynthesis to produce ATP energy, required for active transport, by roots. Alright, yeah, so glucose is required for photosynthesis to produce ATP energy, which is necessary to produce to allow the process of absorption of minerals by active transport. Okay, state the balanced symbol equation for photosynthesis. Now, when we speak of photosynthesis, it is the process in which carbon dioxide reacts with water to give us glucose and oxygen. Alright, so the balanced equation will definitely be carbon dioxide reacting with water to give us glucose and what? And oxygen. Alright, but now we can see that we had 6 molecules of glucose over here and we need to balance it. So we had 6 carbons here, we'll balance 6 carbon over here also. Alright, yeah, then we'll have like 12 oxygen over here and we need to balance 12 oxygen again. Here we have 6. Yes, so balance it here with a 6, and of course here 6 also. So here we'll have like 12 oxygen, 6, 18. Then here we'll have 6, 12, 18 oxygen, and then we have 6 carbons, and of course we have 12 hydrogen also. That is how the equation is balanced. Alright, we move to the next question. Number 5 says the eye is a sense organ that is part of the nervous system. Explain why the eye can be described as a sense organ. Now, an organ, it's a part that contains different cells and tissues that respond or perform a certain specific function. Alright, so we'll say that the eye is a sense organ because it contains cells that can detect light. Alright, yeah, so it is a sense organ because it contains cells that can detect light, and respond to it. Okay, yeah, detect light and respond to it. So they can respond to light which is of high intensity, these cells, but also they can respond to light which is of low intensity. B says state 2 parts of the central nervous system. Now the central nervous system is made up of 2 main parts. A part known as brain, but also a part known as spinal cord. So the central nervous system is definitely made up of brain and spinal cord. C says figure 5.1 is a diagram of a section through the human eye. Diagram of a section through the human eye, as we can see over here. Draw an X to identify a blind spot. Now, blind spot is a part which does not have receptors, that cells that respond to light. So it means it doesn't have cones and it doesn't have loads at all. And this part is definitely somewhere over here. Alright, the blind spots. Okay, then table 5.1 shows parts of the shows some parts labeled in figure 5.1. Table 5.1 shows some of the parts labeled in figure 5.1. Their names and functions complete table 5.1. Okay, so we have the parts, their names and functions, then we have to complete the table. Alright, so we'll start with the first row here. The name of the part, we have to state it, but it's represented by letter H and the function. Now we go back to the diagram and look for letter H. Okay, so that is letter H. From the diagram here, this part usually represents the retina. So the name of this part is retina. Now what is the function of the retina? It contains light receptor cells. It contains rods and cones that respond to what? To light. So we would say that it contains rods and cones that respond to light. Alright? Then we have this second row over here, which transmits impulse to the nervous system. Now the thing that transmits impulse to the nervous part is the optic nerve. Alright, and it's definitely this part over here labeled G. Okay, so it's labeled G, but the name is optic nerve. Alright, then we have the cornea. The cornea usually reflects light. That is the function. It reflects light. Now we have to go back to the diagram and identify where the cornea actually is. It's the outermost layer here, D. Alright, so the cornea is D, and its role is to reflect light. D says, state the names of the two effectors that contract and relax during pupil reflex. Okay, so during a pupil reflex, the effectors that relax and contract are usually the ciliary and radial muscles of the eye. Okay, so we'll say here we have the radial and sicula muscles. Yeah, radial muscles and sicula muscles of the eye. E says, explain why a person is unable to focus on a distant object if the suspensor ligaments become permanently stretched. Okay, so if the suspensor ligaments become permanently stretched, it means that it remains wide. It can no longer become thin. Okay, and if the suspensor ligaments becomes permanently overstretched, it means we will not be able, or the person will not be able to adjust the lens of the eye. Alright, because when focusing a near or a distant object, the lens is changing. Okay, so when you are focusing on a distant object, the lens needs to become a little bit thinner so that we can view an object from far. But when the suspensor ligaments have been permanently overstretched, it means the lens of the eye remains permanently wider. Okay, and if it remains permanently wider, it means the objects that this person can view will only be from a near object. The lens will not be able to adjust so that it can allow light from a distance to focus into the eye. And if such a thing happens, it means this person will not be able to see objects from far. Alright? Yeah. So we'll say here, when suspensor ligaments are permanently overstretched, the lens remains wider. Okay, it can not focus light from a distance into the retina. Hence, a person cannot view a distant object. Alright, yeah, so this is what is happening in terms of accommodation of a distant object. Okay, we are on to question number 6, which says that golden rice is a type of rice that has been genetically modified to contain a nutrient called beta-carotene. Figure 6.1 shows the genetic modification process used to produce golden rice. So this is genetic modification process that can produce golden rice. We can see here we have a bacterial cell, and then we have a plant DNA. We have part of the plant DNA and part of the plant gene. Then they were joined together to form this C over here. C was then brought back into the bacteria and then grown, and then we had a lot of what? Of golden rice together. Okay. Then the question goes on. A says, using the information in figure 6.1, complete the sentences to describe how rice is genetically modified to contain beta-carotene. The part labeled A in a cell is, what is the name of the part labeled A? We come back. So part labeled A is over here. It is inside the bacteria. This part here is known as bacterial plasmid. Alright, so this part here will be identified as a plasmid. Alright. Then the next part says the part A is extracted and cut using a, what is the enzyme that is used to cut the bacterial plasmid? This enzyme is known as restriction enzyme. So we'll state here restriction enzyme. Okay, restriction enzyme forming. Okay, when we cut part of bacterial plasmid with restriction enzyme, we form something that has sticky ends. So here with sticky ends. The part labeled B is the dash that codes for the production of beta-carotene. The part labeled B here is a section of the plant DNA. So this section of the plant DNA coding for this beta-carotene is going to be a gene. So we'll say this is the gene that codes for beta-carotene. The enzyme used to cut part A is also used to cut part B from the plant. So restriction enzyme again. Part B is inserted into part A using the enzyme. Now these two genes will be inserted into each other using something known as DNA regase. Alright, so using the enzyme DNA regase. Yeah, using the enzyme DNA regase. This forms the part labeled C, which is called, now let's see, the part labeled C here. So this part C here has bacterial plasmid and has the gene from the plant. So this thing here is usually known as a recombinant. Okay, because it has two genes from two different organisms. So it's called a recombinant. Alright. Then part C is put into bacterium. The bacterium is taken up by rice plants, giving them the ability to produce beta-carotene. Tissue culture is a form of dash reproduction that is used to produce many identical rice plants are producing beta-carotene. Now it is producing identical rice plants, so they look the same. A form of reproduction that produces identical offspring is asexual. So here tissue culture will be a form of what? Asexual reproduction. Alright? Yeah. Apart from structural features, state two reasons why bacteria are useful for genetic modification. Alright, so bacteria are useful for genetic modification, first of all, because they have no ethical issues. Alright, they have no ethical issues. They don't raise ethical concerns at all. But apart from that, they are simple structures and they are easy to handle and manipulate, and also they reproduce rapidly. So here we say they reproduce rapidly, they are easy to handle and manipulate. Okay, part C says discuss the disadvantages of genetically modifying rice plants to produce beta-carotene. Alright, now what are the disadvantages? Okay, one of the disadvantages is that people have ethical concerns, not widely accepted by consumers. So not widely accepted by consumers. Alright, so they are being produced, but the population that is meant to take them is not very much willingly to do that. But also long-term effects are not known to consumers. Alright, so we say here long-term health effects are unknown. Now this raises a concern to people. Alright, apart from that, the seeds or the products can be very expensive for normal people to purchase. So say here the seeds are very expensive. Alright, yeah, so those are the some of the disadvantages of genetically modifying crops. D says beta carotene is required to produce vitamin A, which is essential for eye function. State the nutrient required to prevent scurvy. Now, scurvy is a disease which involves bleeding from gums. What is the nutrient required to prevent scurvy? It's vitamin C. So we'll state here vitamin C. And then rickets. Rickets is like bending of bones. Alright. So the nutrient that is meant to strengthen the bone is what should be stated over here. And this nutrient over here is vitamin D.